So just for those of you who have not been here before, this is the Tools and Technology Seminar Series. It is a venue to discuss new tools, technologies, and methodologies that are currently in development, recently developed, or just of interest to researchers. If anyone's interested in giving a presentation, either in late May or for the fall, please let me know. And today I'm pleased to present David Shultis, who is going to talk to us about EvoDesign.
Should I kill the lights or leave them on?
No, leave them on, please.
Okay. All right. So I work at the Zhang lab, and I'm based primarily up at the North Campus Research Community and Research Campus in Building 520. So it's great to see some of my 520 guys up here. Today I'm going to be talking about protein design, but this is intimately related to or tied to the process of protein folding. So the Zhang group has two goals. One is to predict the structure and function of macromolecules to aid in understanding sequence to structure and function relationships. And the other is to flip that around, to design macromolecules with altered, modified, augmented, or novel functionality for therapeutics, diagnostics, research tools, and improved understanding of protein folding in general. So most of you are, of course, maybe more related to looking at a lot of DNA structure, a lot of DNA sequences, maybe chromatin structure, and what have you. But sooner or later, your field is going to be inundated with genomic information. And when that occurs, you are going to do a lot of analysis and looking at genes and where they're sitting in these chromosomes and what have you. And then you're going to reach this point at which your informational, ability to extract information from that DNA is going to come to a limitation. And that is primarily because we like to think that most of the biological phenomenon in the cell occurs due to proteins, and that proteins are really the mastermind of driving cellular metabolic processes. If that's the case, then your ability to predict protein function should then extend your ability to understand physiological phenomenon regarding to human disease and treatment thereof. So if you can use functional analysis of the human genome to predict what's going to happen in the cell, this will push your ability to associate genetic code with a physiological phenomenon much greater. And that day is, soon coming. Rob Lyons up at the NCRC has told me that they've actually moved their genomic data recording hardware from Central Campus to North Campus because they're recording so much data. The data transfer rates are now so high that they're now moving everything by hard drive. So that day is coming, and you guys are going to be awash in genomic information. So understanding the processes of protein folding will help you extend your genomic analysis. So I'd like to kind of cap off that by talking about Christian Anfinsen's statement from his Nobel speech in '72, that the three-dimensional structure of a native protein in its normological milieu is one in which the Gibbs free energy of the system is the lowest. And for those of you who don't know, our department chair, associate chair, Gil Oman actually worked with Christian Anfinsen in his younger years. So, very important phenomenon. So the protein folding problem is very complex. We have a simple DNA sequence here, and this is converted into RNA. The protein is then translated by the RNA is then converted into an amino acid sequence by the ribosome, and then the sequence is going to undergo a folding process that gives you incredible functionality based on this very, very unique architecture coupled with sequence specificity. And if it weren't for this incredible diversity, you wouldn't have the wonderful ability to do all the metabolic processes that you do in your body. So this, we like to think of Tom Steitz, who elucidated the structure of the ribosome, said you can't have function without structure. And by that, he was referring to the really complex and diverse functional phenomenon that occur because of the unique shapes the proteins undertake. The problem with this is that in terms of a peptide, this can take an enormous amount of conformations and it can undergo extreme variation in sequence. But if the protein folding process is driven by Anfinsen's thermodynamic hypothesis, and there's been no indication that that's not the case, then we can think of structural modeling and protein design as really the same process, but from different sides of the coin. So we can take a primary sequence, such as the one shown above, and predict its structure by using the iterative threading assembly refinement algorithm from Yang's lab. Or we can turn around and take a structure scaffold or a desired function and then turn around and design a sequence. And really these two processes are governed by the same underlying principles. Again, the real problem is that sequence variation is so large, and sequence space is large, it varies as 20 to the n, where n is the number of amino acids in the polypeptide chain. So very quickly the problem gets out of hand computationally. This is the I-TASSER structural modeling package from Zhang's group. It's won the critical assessment of structure prediction competitions over the last decade. And it works basically like this. You take a primary sequence, you run an algorithm that looks for similar structures and sequences by a process called threading. From this, you get a series of templates out of this library, or out of this search, and then these templates are excised into fragments, and these are used as a series of restraints to do structural assembly. There's about 15, I think, different terms in I-TASSER that are used to drive the structural assembly process besides the restraints, and then we do a process called clustering. So clustering is really looking at the structural similarity between the various structures that were computationally derived, and if there's not close similarity, then in terms of hyperspace, these structures will be very dissimilar, and they would be shown as kind of points here and here in hyperspace. But if the structure is assembled, they'll all be kind of clustered together, and we can use this to help derive what is the most likely, the most probable structure, or the structure with the highest probability of being as close to wild type or native as possible. So this is called the cluster centroid. So these are then these are used to drive new restraints, put through the structural reassembly process, and out of this we do some refinement and get to a final model. Yang also came up with a nice way of looking at structural differences between proteins by looking at this TM score. This is a little bit different than the root mean squared deviation that you guys are used to seeing, in that because you have this DI over DO, where DO is a specific constant, it tends to downweight amino acids that are in dissimilar positions using a C alpha trace. So if you have two different proteins that have the similar fold, but maybe the N terminuses, beginnings of the proteins are wildly different, then this function will downweight the large differences and really focus on the residues of the protein for structural similarity score that are close together. So it's nice for looking at proteins where the fold between the two are similar, but you have certain fragments of the proteins which are highly dissimilar. This is an accurate I-TASSER model that was generated in the Cas9 competition from Vibrio paraholitis, and it was solved by the Northeastern Structural Genomics Consortium. Give you an idea on the ability to predict structures. So these are blind tests, and they don't know what it's going to look like. They don't know what the actual structure looks like until they finish the contest out. All right, so if we can do structure prediction, which is shown up here, we can also flip that around and do protein design. So we're going to talk about now, for the rest of the talk, we'll be focused on designing proteins. This is a survey that Pralay Mitra and I worked on. This is the website that you can access by this, and these are the two papers related to this, and then hopefully I'll be talking about some work that's under review right now at the Biophysical Journal. So the protein design algorithm is a hybrid method. It's composed of both physical and statistical or knowledge-based potentials. Some would say evolutionary. This is the first to use a structure-based protein sequence profile on the Monte Carlo simulation, and it's the first, as we know, that also incorporates secondary structure, torsion angle, and solvent accessibility parameters in the Monte Carlo simulation. It also uses sequence-based clustering techniques for final sequence selection. So a lot of what we learned from protein structure prediction with I-TASSER we're now basically flipping around and we're using the design algorithm. So the design algorithm can be thought of as having three basic parts. First, we're going to generate a protein profile construction, which is much like protein threading. Then we're going to do a randomization simulation, a Monte Carlo search simulation, and then we're going to do a final sequence selection via clustering. How this basically works is we input a target structure, such as the X-linked inhibitor of apoptosis, BER3 domain, shown here with a cognate binding peptide, and we do a search through the PDB library for similar structures. So let's say we come up with, I don't know, 15, 20 different structures using Tamoline. And then we look at these structures and their sequence relationship, and we derive essentially a sequence profile, which is an L by 20 matrix that relates the similarity between the different structures. What this gives you is essentially amino acid probabilities, disease position. The profile at first is not continuous. So to make it continuous, i.e. we can use any of the residues, we actually blur this profile by using a Blossom 62 matrix derived from Stephen Henikoff's work in the mid-1990s to make this continuous. So if you have a tyrosine in the profile, you can look at phenylalanine as well because we have a Blossom 62 matrix to deal with that. So we derive the sequence profile, and then we're going to use this in the Monte Carlo simulation. So the Monte Carlo simulation works like this. We start with a random sequence, and then iteratively we do a series of random mutations to the sequence, and then we score the sequence. And we have two potentials in this scoring potential. One is evolutionary based, and the other is physical based. The evolutionary potential has the multiple sequence alignment, the sequence profile. It's also conserved of single sequence secondary structure predictions or profiles or potentials, solvent accessibility, and torsion angles. We add to this a physical potential, currently using a program called FoldX, in which we look at the difference between the unfolded and folded states and derive an energy score there. We then take z-scores for each one of these scores, which are derived and by comparison with basically random sequences and random folds, and we derive a total score, which is a Monte Carlo energy. And what we do is we look at the difference between, let's say, sequence n and sequence n-1. So we'll always accept into our final sequence stack if the difference between the sequence n and the last sequence is lower, and then there's also a probability based on this distribution of e to the delta e over kt if it's actually less favorable. And this allows us to bounce the sequence out of unfavorable trajectories. So you have to incorporate this if you don't want to get stuck very quickly in a local minimum. And last, what we do is we take the 300,000 sequences from 10 independent trajectories, and we cluster them using the Blossom 62 matrix. And this is done with a clustering size of 40% of the sequences. So once we have that, then we basically, I go to my desk and I convert the protein to a gene, and then I send the gene to a commercial fee-for-service vendor to have it synthesized, and then I'm going to incorporate that gene into an expression plasmid, stick in E. coli, and we're going to make the protein. So this now we can talk about actually taking the algorithm and actually using it to create things. So in phase one, hopefully I'll get to phase one and phase two in our talk today. I'm just talking about designing single protein domains. Not with protein-protein interaction partners, not with an enzymatic function, just designing single protein domains. So we'll work with 9 protein domains to start, and we'll talk about the initial biophysical characterization of these. So how we did this was we had our protein of interest, our design gene here, and then we fused this for expression in E. coli with a series of elements. One is an affinity purification tag, that's 6 histidine amino acids in a row. Another is a solubility domain, the monomeric OCR domain. And then last is a tobacco edge virus protease cut site. If this allows us to purify our protein, this helps give this protein domain time to fold. If this protein is folded, it'll give this time a chance to escape from small kinetic traps to get into. And this allows us to separate all this stuff from this later on. So basically, you purify this entire element, And then you separate this element from all of this by adding a protease, which cuts in this region. What we found was that 7 of our 9 designs were soluble. 1 we couldn't cut the solubility tag off. 1 we couldn't separate out the Mocher solubility tag. 2 didn't express. And 5 were subject to biophysical characterization. We found of those that 3 of the 9 were well folded and stable, and I'm going to show you some of the experimental evidence we have to support that. So this is a SDS stain, or Kamasi stain SDS gel, SDS page gel. It's essentially 4 or 5 centimeters on the side, and the thin axis is about 1 millimeter. What we do is we load our protein samples up here, and then we run voltage over the gel, and then they basically, the proteins basically run down this way based on amino acid length or protein mass. Here we have our molecular weight marker on the left, which is in kilodaltons, and here we have our purified proteins. These are after gel filtration, so these are highly polished, and these are with the fusion tag on. And you can see that we're getting to fairly high purity right now. It's probably greater than 95%, which is good enough for most of the biophysical characterization techniques we're going to use. So this is very satisfactory. We cut the tag off, we remove this stuff, and then we just work with the shorter genes. So of the 5 sequences that we could work with, 1 was from a test set from Mycobacterium tuberculosis, and shown here in purple. And these are 4 other domains that we worked with. These are the PDBs of the target scaffolds that we worked with. These are the lengths. They range from basically 68 amino acids all the way up to 146. This is the sequence identity between the designed fold and the native that we use as the target. This is the type of fold, whether it's all beta strand, whether it's alpha beta, whether it's alpha helix alone, alpha beta, or beta. These are the predicted structures versus the wild type by a TASSER, given an RMSD. And this is whether the proteins were expressed, soluble, they had secondary structure, i.e. alpha or beta strands. And then this is whether they had a stable 3-dimensional fold.
Oh, excuse me, sorry. Love domains in particular fold around a flavin cofactor.
Right.
Are you accounting, I mean, how well is that getting accounted for when you're trying to do this design? I assume that this is going to be an issue with some of the other things also that you may be working.
That's a really good point. So because we
like
have...
they don't even fold without the flavin.
Right, right. So because of the... I will show you what the effect of the structure profile is. And so in regions that are very well conserved, that structure profile is going to really drive those amino acid sequences to that region. So, you know, I'll show you the statistics on that in a second. So you'll see the impact of that. And for the
it ended up not working in this case, but right, the 3D fold is marked as a minus.
Right, right. This is I will show you data that that pushes that on, but that could be a real possibility. So ligand binding and dimerization is definitely a kind of an area, or multi-domain proteins is kind of a next step for us, for sure. Very good question. These are the 5 predicted folds by a TASSER, and here are each one of the protein names. And these are the circular dichrosin spectra for those protein. So here you have the wavelength in nanometers of the incident light, and this is the mean residue ellipticity. So what you're doing here is you're taking your protein sample at really low concentration, and you're sticking it in a cuvette, and then you're illuminating it with left and right-handed circularized polarized light. Is everyone familiar with how that's created? It's a really amazing phenomenon. And the protein, due to its fold, will basically absorb the 2 different circularized light, forms of light, and you'll get a very interesting different spectrum. And so the mean residue ellipticity basically gives you a profile of this difference in absorption. So for a random polypeptide in solution that doesn't have alpha helical or beta strand, alpha helices or beta strands in it, you'll see a value that's very close to 0. But as it begins to adopt a secondary fold, a fold with alpha helices and beta strands, you'll start to see these secondary structure elements come out. This doesn't mean that the protein is stable. It just means that it has secondary structure elements in it. And what you can see is that at around 220, between basically 205 and 222, you have these strong negative lepticity values, which are indicative of secondary structure. Of interest is the thiodoxin fold, which turns out to be identical to wild type. So even though the sequence identity is around 35%, the fold by CD looks essentially identical. And it was really nice seeing this, because I could look through the published literature and see CD spectra from a thioredoxin domain. So with that in mind, we have some idea of secondary structure in the protein. Now we can move forward using nuclear magnetic resonance, MagReg, and magnetic resonance to ascertain the protein fold. This is a simple 1D shot by NMR of proton spectrum, and it's basically cut between ppm of 7 and 0.5. We're just going to focus on the region of the spectra associated with my proton and the spectrum frequency that is in the range of the methyl chemical frequency, or the chemical shifts, if you will. So we're interested in two features. One is that if you look between F and G, you'll see that the feature space in the my proton chemical shift range is very, very kind of bland for the RNPK, but is very, very unique for the thioredoxin. And essentially, magnetic resonance spectroscopy works on this process of electronic shielding. And if the individual electronic environment around each amide proton is very unique and seems to be consistent or within the time scale of the NMR phenomenon, you'll get these very unique features here. And whereas here, these spectra can either be very, very sharp, which indicates that the amide proton is flopping around in solution, or they can be very, very broad or not seen at all if it's working on a conformational exchange or in mobility that's on the time frame of the NMR spectrum. Here are the methyl-chemical shifts, and these methyl-chemical shifts come from basically valine isoleucine residues where the methyl groups for the side chains are well buried. They experience an electronic environment that is very strong, and it shields the proton electrons for that particular nuclei greatly, so they experience a lower NMR magnetic field strength, and thus they have this frequency range that shows up here in terms of parts per millions or chemical shifts. So we see that G, H, and J have these unique features, and we come to the conclusion that these proteins are well folded, while as these are moderately folded. There is some structure there, but it's not. These are not stable domains. So we use two experiments to look at the secondary structure using CD, and then we look at the fold of the protein by NMR. So then, with that in mind, we worked with the thioredoxin domain and the cytokine-independent survival Fox homology domain. We were able to get crystals of this domain. A biochemical rotation student, Greg Dodge, came in and crystallized our Fox homology domain, this PX domain. And he got crystals here in this condition: sodium acetate pH 4, 100 millimolar sodium phosphate, 30% PEG 2000, and down here, and the longest length is about 300 to 400 micrometers, and on the short axis they're about 50 or so micrometers. So these are very, very small, and you can't really see them by eye, and you need at least like a 10 to 100 pound power microscope to see them. So we took these crystals to the Advanced Photon Source at Argonne National Labs, synchrotron, and we illuminated them with 1 angstrom, essentially 1 angstrom light. If you take a protein crystal and you put it in in this beam of light, you get about, I think, 10 to the 13 photons per second. You can get a diffraction pattern, and this is essentially a charge-coupled device. It's a plate. We put the protein crystal in front of the beam. The beam of light is non-divergent, meaning it's kind of like light from the sun, and it comes through a narrow slit that's about 1/100th of a square millimeter, and this light impinges on the crystal, and then the atoms within the crystal all resonate elastically, and you get constructive interference patterns. These interference patterns show up at these little reflections, which are called Bragg reflections. The angle between the center right here and the going outwards gives you an idea of the resolution of information that is in the data set. This beam coming across right here is simply a beam stop that keeps the synchrotron light source from destroying the detector. This ring right here is a water shell that represents about 3 angstrom resolution. So as you go out here, this gives you increasing information content. These are a series of 3-dimensional waves that you add up to form an electron density map in a box. You can think of this as a shoebox, right? And each one of these spots represents a 3-dimensional wave, and this is the magnitude of the wave. The problem is that it doesn't give you the phasing information in that wave. So we know that a wave has a frequency, it has an amplitude, and then it's going to have some phase information. The problem is X-ray crystallography does not give it, currently using this technique, does not give us the phase information. We rotate the crystal through basically 1-degree oscillations. You collect around 300 data frames, and you determine the basically box that the protein sits in, which is known as the unit cell, which then derives like the symmetry operations that allow us to basically reconstruct the protein within that box based on the space group assignment, which is known as a primitive orthorhombic space group. And this tells us that there are basically 4 molecules within this unit cell. There's about 1 trillion molecules of the protein within these small protein crystals, which is an amazing number, right? 1 trillion proteins are used to form the crystal that led to this diffraction pattern. The reason we use protein crystals for this is that the crystals amplify the signal. Now people are starting to use free electron light sources to basically vaporize proteins and get single molecule scattering patterns. But this is currently the old school and traditional way of collecting structural information using short wavelengths of light. So this is our statistics behind our data collection set. These are the actual unit cell parameters. This is the basic box that makes up the information that we use to model our protein in. And this is the resolution range. We collect probably around 70 or 80,000 of those spots we call Bragg reflections. And then we work this down to about 12,000 reflections by noting that many of the reflections are redundant. They're the same data set. We split this into a working set and kind of a test set, just like you do all your computational information training. We look at the completeness of the set versus the possible set size, and we look at its multiplicity and signal to noise. And there's some other data that may be of interest to other structural biologists. Overall, when we refine the structure, we found that the favored regions for the Ramachandran plot for the dihedral torsion angles is quite good, and there are a few in the allowable regions, and they have to do with some loops that we had difficulties modeling with. But overall, this is a very good data set. It's complete, and it has high signal to noise. So these were the very first electron density maps shown on the right. I fitted the basic C-alpha model to give you an idea of how the helices sit in this protein. We developed this phasing solution using molecular replacement in the original model. We used the original Fox homology domain model to derive our phasing information that we don't get from the X-ray crystallographic experiment. We have some statistics to tell us whether that's correct or not. We knew that this was a very good solution right away. Here you're looking in blue is your electron density. This comes due to the scattering of the electrons associated with the atoms. Right here you can see amino acid side chain. This is most like a phenylalanine. And then here you have a little bit of the backbone and another helix nearby. So the green elements represent difference that isn't accounted for in my structural model, and the red indicate regions that the algorithm is telling me they shouldn't be there. And then we have ways of controlling the signal to noise, so you can make this much stronger or weaker depending on your cutoff. You can filter that out. So we built this model, and after refinement, this is the electron density we see around phenylalanine 45. You can very clearly see the aromatic ring structure, and down here at very, very low, you can actually start to see the middle of the ring structure, so that the electron density actually gets very weak there. So now we'll move to more structural analysis. We're going to compare our designed X-ray structure, our design domain X-ray structure, with the wild type. So the design is always going to be in blue, and the native is in green. And what you see it has, I think, 3 strands and 4 alpha helices with a number of loops. There was also this sulfate anion that was bound in the crystal structure. It turns out to be quite interesting where that's located. These loops down here were hard to model. Electron density is very poor there, so I'd just like to point that out, that this is a very, very poor fit. But these happen to do with a large number of positively charged residues. This is the match between, in blue, the X-ray crystallographic structure of our design domain and the I-TASSER structure. I've kept the sulfate molecule in for the design domain in the X-ray structure, but I-TASSER does not have that. But because it's using a threading-based template model, and we're in real mode, it has a high chance of predicting the structure because we've used a structure profile in the design simulation. So many of the features that I-TASSER recognizes in terms not just of the threading templates, but also the statistical potentials show up really well. So I-TASSER is actually able to fit this structure well at about 1.32 angstroms difference RMSD between the design X-ray structure and the I-TASSER model. Over here, the question is, so what does this domain do, David? What's the role of the cytokine-independent survival kinase Fox homology domain? And this PX domain is actually fused to the cytokine-independent kinase domain, and it's involved in localizing the kinase domain at the membrane. Remember, it has a structure profile which is helping drive certain key features of the protein fold in the design simulation. It also turns out that profile is also driving amino acids that will be associated with membrane association. So this domain fuses, likes to bind, it's a peripheral membrane protein, and it likes to bind to lysosomal compartment phosphonosides. And there's specific phosphonosides that this design, or the protein likes to bind to. It turns out because of the structure profile used, there's high similarity in this. In fact, the sulfate molecule binds in a similar way that the phosphonositide does in a similar Fox homology domain structure I'll show you in just a second. So this is the sequence alignment between the wild type and the design structure, where the strands are represented as arrows and the helices as ovals. And the purple represents sequence identity of 100%. So to get back to your issue is how does the structural profile work with design domain and what's its impact? We can look at a Shannon entropy map. Claudius Shannon came out of here at the University of Michigan, and then he went to MIT, and he's considered the father of information theory, right? You guys remember that at all? Yeah. So he came up with this idea of entropy, and he was talking to another physicist, and he was like, oh, you should look at this guy Gibbs's entropy. And so he used that model to describe his own variation of entropy. And so he came up with Shannon entropy, and this reports on the log probabilities for each amino acid at each position. And dark red indicates low entropy, i.e. very well conserved. And then dark blue represents high entropy and not really conserved. Down at the bottom is the summation, the total entropy for that position. And you'll see that in both the wild type and the design, where there's strong entropic, the entropy is quite high. The match is quite good, and where the entropy is low, the match is quite good. Particularly, if you look here in these red segments here, you notice that in some cases, the sequence conservation is more kept with the design than the wild type. And so there's a sulfate that I've spoken about for the design that shows up in the crystal structure, and it binds to these residues shown right here, and you can see this in this structure below. The residues involved in the that are very well conserved in this map show up here in red. And so these residues really help drive the fold and thus the function of the protein. So this has the tendency of giving us a more, for a given function, we're going to have a greater probability of being successful, I believe, in our design simulations if we have this structure profile with us, because Mother Nature's already mapped this space out. So we can use this for our design in the future if we want to design a protein that binds a specific phosphoinositide for use in vivo assays for research, diagnostics, or therapeutics sometime in the future. So one of the interesting things is, so if the structural profile is going to, look at the entire fold space for all these Fox homology domains, what's the impact on it? So here in green, you can see this loop from the wild-type design protein, and this is another structure of another wild-type, another native Fox homology domain, and there's a proline here. And this proline shows up in our design as well. And this proline really restricts the motion of this portion or sequence of the amino acid chain. And so you get this angle of the design domain that follows more closely this other Fox homology domain that was not the target template. And this is important because even though there's variation in this region, a lot of the statistical potentials are helped drive in the amino acid selection so that it doesn't get too far off the beaten path. For instance, if this part of the protein only bound to another part of the protein up here in green, and suddenly you put this proline in here, well, maybe the whole thing is not going to work that well. And so by using the statistical potentials, there may be some features that are encapsulated into the sequence selection region here that are, that help out the general process. I.e., what I mean by this is that a physical-based potential, as soon as you vary the structure, right, you have to account for that. And that's going to be next to impossible using standard physical-based potentials because you've actually fit this model to here. So it's not going to sample that. And you can't sample all these different conformers for every amino acid change you're going to do in the design simulation. So we do 300 cycles for basically one fixed structure. And if you start moving that structure around, that means you have to do, for every move, you still have to do those, all those mutations. So it gets really, really hard. So here I think is a benefit of the structure profile. And the bottom line is I think it basically keeps you on track. So on here is this P40 Fox homology domain with a phosphoinositide analog bound in the X-ray crystallographic structure. I have taken that phosphoinositide and by superposition put it in this region where the phosphide anion is sitting in our crystal structure, and the likeness is very, very high. There's no specific clashes, and the RMSD between the two phosphate groups, between the crystal structure shown here and here, turns out to be less than an angstrom. So they sit very, very similar. If you look at the residues here, you have arginine 58, arginine 41, histidine 38, histidine 22. These residues are well conserved in the structure profile, so they show up in the protein. What you notice, though, is the binding pocket is a little bit smaller here. It's larger here. These are differences that are not conserved between the individual domains. So we still have room to, we can drive, let's say, if we want to make a binding profile and incorporate it in the design engine, we can still mutate this around and still keep the possibility of binding a phosphoinositide, but have different features. We did not measure the binding of this phosphoinositide to the design domain. So how did if we do a kind of we step back and we look at all the secondary structure, solvent accessibility, and rotamer errors in the design in comparison to the wild-type domains, we can kind of look at maybe the impact that these potentials had in our design simulation. So up here we have a secondary structure given by Yang's PSS pred, and in blue is the wild-type protein where 1 is a perfect match and 0 is an error. And then in red we have the design sequence. And it works out that the accuracy by PSS pred for the design simulation is 91%, and for wild type is 86%. If we go back and reanalyze this by EvoDesign, the difference is definitely skewed. So EvoDesign is recognizing the design sequence with a statistically significant difference, as well as when we look at the solvent accessibility. So we predict the solvent accessibility for each residue, and we see that in general the solvent, which is another program that Yang wrote, as minimal differences, but EvoDesign definitely recognizes the difference. And so you're definitely skewing the outcome in favor of amino acids that the EvoDesign secondary structure potential and solving accessibility potentials recognize. If we look at the rotamer error, this is very unique because we're using the side chain fitting program Squirrel in this. This difference is definitely significant, and it's about a 10-degree difference spread. So you're just predicting the angles for the side chain rotamers. So overall, we know that each of the potentials in regard to EvoDesign is definitely recognizable. And these are helping guide the selection of amino acid sequence that has a larger, or we think more likely, probability of actually working. So to wrap up, we just say that we've designed a molecule with atomic accuracy, and that we've been able to predict that structure with I-TASSER. So that's our kind of our first our phase, and now we're going to switch to designing functional proteins. We have probably about 10 more minutes. So I'll just kind of get into a little bit of the background behind this, and then I'll get to the data that we do have, or some of the data. So we're interested in starting to design functional proteins now. We're going to focus on protein-peptide interactions, and here we have a map of the basic biochemical pathways associated with cancer. We're going to focus on this protein called X-linked inhibitor of apoptosis, specifically a BER3 domain within this. The BER3 domain is known to inhibit Smac proteins and inhibit caspase 9. So this protein called Smac or SMAD binds to caspase 9 and basically, excuse me, the Smac or SMAD will bind to XIP and it will block the XIP caspase 9 interaction. When caspase 9 is free, its normal activity is to start the process of apoptosis. There are other controlling factors involved, but this is one of them. And so we decided to design an XIP BER3 domain that would bind a Smac peptide, and we can use that as a research reagent in cell-based studies. We're actually trying to incorporate these functionally designed proteins into in vivo cells in a breast cancer cell line to see if we can perturb the system. So this is kind of an important work that really hasn't been, I think, examined much by the protein design field, which is when you stick these design domains back in a protein, in the cell, what do they do? And I kind of liken this problem of you'd like to be able to perturb the system in a way where you still have, let's say, one design function, i.e. you design the XIP to bind the SMAD, and that would basically keep this wild type caspase 9 function intact, so you're not causing a cell to die. And so you could use that in the presence of compounds that actually drive the cell to apoptosis, and you could tease out various biochemical pathways by the incorporation or addition of these design domains. And what's nice about this is that they may be silent in the cell, i.e. their outside surface may be completely different to a large degree other than, let's say, the functional binding epitope. So they may not be inhibited at the RNA level, they may not be inhibited at the gene silencing level, and so you have ways of kind of overcoming this in ways that the normal cell is not going to respond because it doesn't think that you're putting in an XIP domain in terms of biochemical feedback loops. That may have an advantage later on. So we're trying to look at that. The idea is you basically target biochemical pathways that are under active drug discovery, i.e. that are in phase 1, phase 2 trials and help ascertain, what's going on. So these were not only just the XIP structure, but we were also targeting other proteins associated with this. We've been successful in designing XIP domains and also as well as PDZ domains. I'm just going to talk about the XIP domain with the SMAD-SMAC peptide. So XIP, the BER3 domain, binds the N-terminus of this protein called Smac or SMAD, and it does so at a dimer. And when it does, the normal inhibition of XIP of caspase 9 is blocked. So what you have is Smac, which is released from basically dying mitochondria, is going to bind competitively with XIP, and it's going to block this inhibition. So a negative of a negative gives you a positive, and this caspase 9 goes active, and it turns on the programmed cell death pathway. So that's what we're going to be targeting. Our design is our focus right now is to see if we can design the XIP BR3 domain. So in blue is the wild type structure, and in red is the predicted structure by I-TASSER. We had a designed interface, and then we also kept the design, we modulated the design, but we kept the interface fixed for a protein we call the fixed interface XIP. And we did this as a control because we really, we had no idea of what the outcome was going to be. It turns out the outcome is good for both of the designed proteins. They both bind the target peptide. So here we have, for the designed interface, we have mutations in this interface that are different from wild type, and there's about a 50% sequence variation from the design. Down here you have the secondary structure elements of the protein, and you have the wild type XIP, the fixed interface, or the designed XRPs. This is where the peptide binds down here, shown in these black dots, and this is where the caspase 9 interface is. The caspase 9 interface is much larger. So first we did the same thing we did previously with our monomeric domains. We looked at the protein folds by circular dichroism. Here's your wavelength down here. Here's your mean residue ellipticity here. And all three of the proteins have strong negative features. These proteins, though, the fold space is very, very unique. It's very, very strong here. And I don't know how to analyze this other than to say that they do have some secondary structure elements in them. That's very, very unique. We then took these proteins and put them in an NMR tube and collected the protein spectra, and this is the full spectra now that I'm showing you. And we did see very, very solid chemical shifts for each of the proteins, for the amide protons, as well as some of the methyl groups that show up kind of down in this range right here. So that was good. We then looked at the free energies unfolding. So we went back to the circular dichroism spectra, but this time we added in a denaturant. This is the denaturant composition using urea and molarity. And this is the mean residue ellipticity for a given wavelength. I think we used like 220, 223, it's pretty high. And what you can see is that the mean residue ellipticity at 0 molar concentration is very low. It gets down to very negative numbers. And then as you add in the urea, it starts to become much more positive, much like you have a free peptide in solution. And from this, we can determine a free energy unfolding, which for a wild type XCP was minus 2.7 kcals per mole, as well as the DI, and then the FI is 2.3 kilocals per mole. This difference between the DI and the FI is statistically significant. There is no difference really between the FI and the DI through statistics. I don't know why right now the FI domain is not as stable as the other two. So we also, in a collaboration with Tomek Czerpiecki's group, in the Department of Pathology, we subject our protein to two-dimensional NMR spectroscopy. Here is the amide proton frequency range. And we've also added an additional element of nitrogen spectra. So we've isotopically labeled our protein. This is a heteronuclear single quantum correlation spectra, or HSQC. This unique feature map here is derived from the amide nitrogen amide proton pairs, and you get this whenever the protein is well folded. So in red are the dots where there's just the XIP alone, and then we titrate in a Smac peptide, which is the short N-terminus that's responsible for driving caspase 9 inhibition, and we titrate it in green and then fully exhaustively to saturation in blue. And we do that for the design interface as well as the fixed interface. And what you can look at very specifically, if we look here over for the fixed interface, you can clearly see that in the Smac, with no Smac in here, you get this red peak here. Then you add in a little bit of the Smac, basically basically 1 to 2 Smac to XIP, and you get kind of green here and kind of green here. And then when you saturate XIP with Smac, you get this blue dot here. So this is telling us that the electronic environment around XIP is changing because the Smac peptide is binding the protein. And this is true for both the design and the fixed interface. So then we subject the protein to isothermal calorimetry with the addition of the Smac peptide, and we found out the dissociation constants for the wild type as a control comparison was 80 nanomolar. The DI-XIP was 167 nanomolar, has much larger variance, and the FI sample was 273, kind of going along with the difference in stability that we saw from the enfolding experiments. And this is kind of the best example we had of our DI-XIAP binding isotherm that's been derived from isothermal calorimetry. So this tells us that the peptide is binding the protein, and the affinities between the two are roughly, are very close to being next to native. So they also looked at, all right, so you've designed an interface that binds Smac. Well, what about, you know, binding caspase-9 and inhibiting caspase-9 function? So Xiaoman Wang's group has a very nice experiment where they can look at the protease activity of caspase-9 in the presence of XIP. And you can add in compounds that inhibit XIP for their assay, or we can just compare the wild type versus the fixed interface and design interface. If the fixed interface or design interface don't interact with caspase-9, we should expect full activity. However, if wild type XIP interacts with caspase-9, we should see a definite loss in protease activity. And what you see in terms of fluorescence scale is that indeed the wild types, you add in more of the protein, you get less proteolytic activity of caspase-9, whereas the DI and the FI samples are unchanged. If you look at this as a percentage of inhibition of caspase-9 activity, here's your basic plot, and there's a very strong difference between the two of them. So we've designed the proteins to bind the Smac peptide, but because of the differentiation between various caspase-9 structures and the large surface area of the caspase-9 XIP BR3 domain interaction, this doesn't carry over. So we were able to get crystals of the fixed interface XIP with in collaboration with the Li Lab with Bingbing Wan, and they've now moved back to China. But they weren't able to get a structure on this. I looked at some of the electron, the diffraction patterns, and we've tried processing the data, and we really couldn't work with this too much, which is unfortunate. And with that, I think we can probably, because we're running short on time, I'm just going to stop right there and say that I'm currently developing a method to use mass spectroscopy to look at the fold of the protein, and there's several people working out with that. So with that, I'll just kind of close up and say that these are all the wonderful folks that have helped us out. I'd like to thank the DCMB, particularly Brian Anthony and Gil Oman, for all the startup money, the NCR space, and financial support for my own stay here at the University of Michigan. Yang Zhang, he has several NIH and NSF fellowships. He's been wonderfully supportive, and all these people here have been helping me out in various ways associated with both of these projects. And then the UROP program has been very supportive of me. I've worked with over 42 undergraduate students. We received 4 undergraduate UROP fellowships, and then these are a list of our collaborators. Additionally, Laura Sessa from Jason Jiswicki's lab. Jason's now moved to UCSF, but I'm still working with Laura, and she's trying to incorporate our design proteins into a breast cancer cell line so that we can look at how the XIP domains can be used to perturb the biochemical pathways for apoptosis relative to compounds that the Xiaoman Wang lab group has for use for cancer therapies. Thank you so much. I hope that I kept everyone awake and everything else. Is there any questions? Okay, Bill.
All right, well, so I did a question early on in the simulation part of your design when you're introducing mutations. First of all, I take it that's a fairly computation intensive thing to do, or is it? If it is, I was just curious. You'd mentioned shortly after that you, and various times, that you're finding proteins where the fold looks very similar to the wild type, but the sequence conservation is not really all that strong. So I'm assuming that means that there are parts of the sequence that matter more to the fold in the sense than the rest. Do you use that to inform which mutations you introduce where, or do you just sort of do it at random and let the method do what it does?
I think it's important. What you can do is you're talking about being smart in the way you introduce mutations. And I think that your idea is right. One of the things, and I think we've actually implemented this now, is that you make you can look at the fold of the protein and say this is a buried region, this is solvent accessible, and you know what those amino acids are, what they should be statistically, so you can incorporate those. But if you start doing more than that, you start really driving the system in ways that maybe you don't want to go. So I think you can do a limited version of smart amino acid selection. Maybe that can be driven. You could use the probabilities from this map to help drive that, but you have to be careful that you really don't limit your search too much. So, where would we like to go with this in kind of big picture? We'd like to start designing protein-protein interactions and start designing antibodies, and then using these to aid in crystallization, using them for basically therapeutics. You can design proteins with very, very high melting temperatures that you can, take out in the field in Africa and assay certain things for. They can give very, very high shelf life, very long shelf lives. We can even potentially incorporate synthetic amino acids into our design algorithm. So right now, this whole program is basically designed this basic engine. Can you make an engine? Can it more or less work? And then over time, you will then fill in, add different features to the design work engine. So far, it seems to work, but not all the time.
So how do you determine the weight in your energy function?
You mean overall?
Between evolution and physical potential.
So by scoring, Pralay worked this out, and he looked at using the feed out is really I-TASSER. So you basically fit this scoring potential, and you look at how I-TASSER thinks about some of these sequences. You've really hit upon a really big problem. It's just how do you train your algorithm, right? How do you weight the scoring functions? And this, we have very, very limited data on this. And this is actually, since I've been in the Zhang lab, one of the things I realize is that the most important thing we can get is very, very large data sets where we understand the impact of mutations and insertions and deletions on that protein. And then you can use this information in your bioinformatic programs to do what? To help improve your functional annotation of the human genome, right? So this basic work and the problem you hit on is one that you can use not only in protein design, but in protein structure prediction, and thus human genomic, functional annotation of the human genome.
Thank you.